Hi, everybody. My name is Ed Davis. I'm Senior Application Scientist at Genecopoeia Incorporated, located in Rockville, Maryland, just outside of Washington, D.C. I'd like to thank you all for joining us for today's webinar entitled Genecopoeia CRISPR Genome Editing Technology. We really appreciate it. Before I go into the seminar, I just want to go over a few practical matters. Excuse me. First, you may ask me questions at any time. But in order to do so, you will not be able to speak vocally to me because your microphones will be muted. Instead, you'll need to type your questions in the questions box located in your control panel. If you can't see your control panel, it might be hidden, in which case look for a small strip of buttons on one side of the screen. One of those buttons will be orange with a white arrow, and if you click on that button, you'll then be able to see the control panel and your questions box. Secondly, in the interest of keeping the talk going smoothly, I will wait until the end of the talk to answer your questions in the order in which they were received. And when I do so, I will speak the question out loud so that everyone can hear. And I will also speak out the answer aloud so that everyone can hear. And finally, I will be sending you a number of links to download various literature like technical notes, application notes. And to do that, I'll be using the chat box, and that's also located in your control panel at the bottom. All right, so with that, let's get on to the seminar. All right, so I'd like to start off by telling you who we are at Genecopoeia and what we're about. Essentially, we are a company that's that markets and provides products and services for functional genomics and cell biology. So one of the main product lines that we offer is expression-ready clones, shown here in this box. And these clones come in a number of categories, such as ORF. This is actually how we were founded, with a large collection of ready-to-express human and mouse ORF clones. These are, again, you can put them into your cells. They're designed mainly for cell lines or bacterial expression. You can put them into your cells the day you get them from us, and you can express these clones with various tags or promoters, what have you. But we also have a number of other clone types, such as promoters. These are regions upstream of the transcriptional start sites for each human and mouse gene. They're intended for mammalian expression, as well as microRNA clones. This includes microRNA 3' UTR targets for each gene, as well as microRNA precursor clones and microRNA inhibitors. We also have CRISPR clones, which you're going to hear about today. sgRNA clones, if you are interested in more traditional knockdown type of experiments. But we've also expanded out into a number of other product areas, such as viral systems. This includes lentivirus. This is extremely popular, so we provide lentiviral-based clones. We have services where we will produce lentiviral particles for people, as well as various kits like packaging kits and cell lines for lentivirus. And very similar is true for adeno-associated virus, or AAV. In addition, we have a very large selection of kits and reagents, primarily for transfection, like transfection reagents. We have a large selection of luciferase kits for both Firefly and the secreted Ghostia luciferase system. Fish probes, fluorescence in situ hybridization probes for various applications. Indel detection, this goes along with our CRISPR products, and cloning kits. And finally, we have a category called fluorescent detection. This is where we have products for cell function assays for things like detecting apoptosis, for example, nucleic acid detection, so DNA gel stains, cell structure probes. These are basically organelle probes, mitochondria, Golgi, lysosomes. And finally, we have a large selection of fluorescent dyes. But of course, you're here to listen to me tell you about CRISPR, so let's go into that. All right, so here's an outline of today's seminar. First, I'm going to go over genome editing as background, talk about the technologies, what technologies there are. Then I'm going to discuss applications for genome editing, all the cool things that you can do. And finally, and most importantly, how we at Genecopoeia can help you with your genome editing applications. So let's start off with talking about the technologies. So for one of the biggest applications for genome editing, CRISPR mainly, is to reduce gene expression or ablate gene expression. Now, for many years, this was kind of difficult to do in mammalian cells, um, especially if you wanted to knock a gene out. But then along came RNA interference or RNAi, and that proved to be a very powerful method for ablation of gene expression. However, again, CRISPR is useful for ablation of gene expression as well, but the two approaches are vastly different. And a lot of people don't realize this. They'll either confuse RNAi with CRISPR and say, for CRISPR, they want to knock a gene down. But actually, while you do get knocked down with RNAi, and of course you're lucky if you're not lucky, but you're doing well if you get 90% knockdown. With CRISPR, the goal is usually to knock the gene out. And again, we're only talking about gene ablation studies. And so that way you can completely get rid of your gene product, which, in the case of a knockdown, 90% or 70% might be good, but you still have some gene product left over, which could confound your results. And it does this by causing CRISPR does this by causing a permanent change in the genetic code. So it will either delete some base pairs, some information, or you might change a base from one to another. Whereas with RNAi, this of course does not happen. It primarily works at the post-transcriptional or post-translational level, but the genetic code is not altered. So this is very important to keep in mind when thinking about how you're going to use CRISPR, how you're going to be doing your screening, because again, the two technologies are vastly different in how you will approach your experiments. So that was a very brief overview, but if you want to know more about the differences between CRISPR and RNAi for gene ablation studies, you can download this technical note from us. This is the download link, but I don't expect you to memorize that. Instead, I will copy the link and paste it into the chat box, and then you can just download it at will. All right, so I just sent that link to you in the chat box. All right, so now let's talk about how CRISPR actually gets the job done. So most CRISPR applications, and this is also true of TALEN and earlier technologies like zinc finger, start off by creating a target site-specific double-strand break, or DSB. So these are essentially nucleases. They're chromosomal nucleases, and they've been adapted from usually bacterial sources or plant sources to do work in other systems like mammalian cells. So when you target a CRISPR to a chromosome, you get a double-strand break, so it cuts both strands of the chromosome. And then this break has to be repaired or the cells will die. And it does so by using primarily one of two mechanisms. One of these is known as homologous recombination, also known as homology-directed repair. And this is actually the more natural form of DNA repair. It's what occurs in meiosis or when you have DNA damage. And so in this case, you get repair of the double-strand break using a homologous chromosome. Okay, again, like in meiosis or DNA damage. However, you can also add an exogenous piece of DNA, which is known as a donor. And this can come in the form of a big plasmid, or it can even be an oligonucleotide. But the whole point is the mechanism is essentially the same. So when you have homology to the sequences on either side of the break, then you get repair of this break by copying information from the donor to the chromosome. And so what people do with these donors is they will insert DNA desired DNA sequences, which can be just about anything, like a fluorescent marker or a particular gene they want to knock in, as it's called, or a base pair mutation. And you get this copying from donor to chromosome, and then what you're left with is a chromosome now that has your new information knocked in. Okay, and this can be used for many, many different applications that you'll see soon. However, in the absence of homology, and this is what usually occurs like in non-dividing cells, if you don't add a donor DNA, you get the cell uses another process called non-homologous end joining, or NHEJ. NHEJ is simply sticking the two ends of the chromosome back together. Now, unlike homologous recombination, which is a relatively error-free process, or a very low error process, NHEJ is a very highly error-prone process. So what you often will end up with is you'll get what's known as an indel, okay, which is an amalgam of insertion or deletion. And these insertions and deletions can range in varying sizes from one base pair up to hundreds of base pairs. And so when these indels occur in the coding region of a gene, of a protein coding gene, you will often, at least 2/3 of the time, get a frame shift mutation, which will then knock the gene out either by both frame shifting, knocking the gene out of frame, the translation out of frame, or it'll destabilize the message by nonsense-mediated decay. So this NHEJ mechanism is a very convenient way, very highly efficient way of using CRISPR to knock genes out, getting complete knockouts. All right, so CRISPR actually comes in a number of different forms. The most popular form used for genome editing comes from the CRISPR system from Streptococcus pyogenes, the same bacteria that causes strep throat, because most bacteria and most archaea actually contain CRISPR systems. There are systems that are used by bacteria for defense against transposons or viruses, phages. But in the case of genome editing, genome engineering, the most widely used form is from Strep pyogenes, and it's a two-component system. One of these components is known as a single guide RNA, which is actually a synthetic fusion of two different RNAs, one which provides structural scaffolding, not activity, but structure to the guide RNA, and the other, known as the cRNA or CRISPR RNA, provides target site specificity. So part of the CRISPR single guide RNA is a 20 nucleotide target site. And actually this target site can be anywhere from 17 to 20 nucleotides, but most commonly it's 20. So the other component of this system is the Cas9 nuclease, shown by this yellow shape. So when you get this complex form between the Cas9 nuclease and the single guide RNA, you then create an active nuclease, okay? So when this occurs, the active nuclease is guided to this 20 nucleotide sequence found in the chromosome. And when this sequence occurs upstream of what's known as an NGG PAM site, it binds to the chromosome, creates a double-strand break, and that DSB is repaired by the mechanisms of non-homologous end joining or homology-directed repair that I mentioned before. So the Cas9 single-guide RNA complex makes this double-strand break 3 to 4 nucleotides upstream of the PAM. All right, so now CRISPR has been a real genuine sensation over the past 3 to 4 years now, and it's still hot. People are talking about it all the time. It's even mentioned in the popular press. I even saw an episode of a TV show recently where they were, it was The Blacklist actually, where they mentioned CRISPR. And I thought that was kind of amusing. But anyway, CRISPR has literally taken the world by storm. It's huge. And that's because it's so efficient and it works very well, and it's relatively inexpensive. However, as great as CRISPR has been, though, it does have one, at least in the form, in its most popular form that I just showed you, has one significant limitation, which is its specificity, or propensity to cause off-target mutations. And that was originally shown back in 2013 by Keith Young's lab, where they found that they could mutate each position of the single guide RNA. That's shown by this graph, each of the 20 positions. And when they did this for 3 different single guide RNAs that were targeted to a GFP site, they found that the activity of the ability to knock out the GFP was still very high. In some cases, it was still near 100% activity when they changed a base in the single guide RNA. Now, there was some variation when you did this, when you created these mismatches. You did get a loss of activity at some positions, but it was still very high. Okay, so this suggested that that CRISPR can tolerate mismatches between the target site in the chromosome and the single guide RNA and still cause cutting. And when they also tested this for activity against natural chromosomal targets, they found that the level of off-target mutagenesis, that is indel formation, was extremely high. It ranged from 5 to 125% of that found at the intended targets. So this created a lot of concern initially that CRISPR may not have the required specificity that people want for certain genome editing applications, especially gene therapy, which is, of course, something that a lot of groups want to do. And now I'm not going to go into all the data about this, but since then, while CRISPR off-target mutagenesis is a concern, it's probably not as big a concern as was originally shown in this paper. People found that the level of off-targeting is actually quite a bit lower in general, and there are a lot of software algorithms that can help you predict single guide RNAs that are less likely to go to off-target sites than others. However, given that, there's still a concern with off-targeting for CRISPR. So in order to address this issue, a number of different technologies have popped up in the subsequent years since this first finding that have been designed to mitigate the level of off-target by CRISPR. One of these is known as a double nicking strategy. This is taking advantage of a mutant of Cas9 that can only cut one strand rather than both strands of the chromosome. And so when you have 2 of these mutated Cas9s complexed with single guide RNAs that target opposite strands on either side of the break, you can then get cutting and repair by NHEJ or HDR that I mentioned before. Okay, and again, this is known as a double nicking strategy. And the nice thing is that these nicks are repaired at a much lower frequency, than double-strand breaks are by the standard mechanisms. And so what these groups have found is that this double nicking strategy dramatically reduces the level of off-target mutagenesis. It doesn't go to 0, but it is a large reduction. Okay, but even more promising than the double nicking strategy has been the more recent development of what's known as Cas9 point mutants. This is a technology that was developed by 2 different laboratories independently. That would be Feng Zhang's lab at the Broad Institute and Keith Young's lab at MIT. And the key results of both papers are shown here. So what these 2 groups did, they independently isolated they created a number of mutants in Cas9 that they predicted would actually bind to the chromosome. It would destabilize binding to the chromosome, and the hope was that it would cause a reduction in off-target activity. And so the mutants that the 2 groups came up with do not overlap at all. So what they found is that when they compare wild-type Cas9 to mutant Cas9 in its ability to create indels in the chromosome, they found that the mutant Cas9s were very similar in activity. The mutant activity is shown by the blue bars, but when they examined off-target activity, they found that the level of off-target cutting by the Cas9 point mutants was reduced to background levels. And the gray bars here represent off-target activity by wild-type CRISPR. But you can see almost no activity, off-target activity from the wild-type, or excuse me, the mutant Cas9s. And a similar result was obtained from Keith Young's group. So this technology, these Cas9 point mutants, seem to be the most promising technological development for mitigating off-target mutagenesis by CRISPR, because the level of activity, of on-target activity, is nearly as good as wild type, and yet the background level of off-target mutations is near zero, or effectively zero. All right, so if you want to know more about dealing with and taming the off-target mutagenesis activity of CRISPR, you can download this technical note. And again, I will copy and paste the link to that in the chat box. Sorry, I'm having a little trouble here, but I'll get it. Okay, and I just sent that technical note link along to you. And if you don't see it, you might need to scroll down in the chat box, but it is there. All right, so now that I've given you an overview of genome editing, of CRISPR technologies, let's talk about applications for genome editing. There are many applications you can do. Some of the most popular applications are shown here, though not all of them. Gene knockout, as I mentioned, is a big, widely used application, and this can be done either by, and these are some published examples. They're pretty old now, but these are some early published examples of using CRISPR or TALEN for knockout of genes in either cultured cells or animals. There's also correction of disease mutations, such as this example where a group cured heritable cataract disease in transgenic mice. There's engineered disease resistance. People have used genome editing tools to knock out the CCR5 gene to cure patients of HIV. That's actually in clinical trials right now. There's forward mutagenesis screens where you can use CRISPR single guide RNA libraries to do screening for like drug targets, for example. In-frame fusion tagging, you might want to add a tag like a V5 or a GFP tag to one end of your gene. And there's also transgene knock-in, such as using the safe harbor system for humans or mice to insert transgenes in various situations. All right, so let's talk first about gene knockout. So what happens when you use CRISPR to knock a gene out? Well, this example that I have here shows what happens when you do a knockout via non-homologous end joining. Okay, and remember I mentioned earlier that in these cases you get insertions or deletions of various sizes, and these are random. They're unpredictable insertions or deletions, so you never know what you're going to get beforehand. And when this is from an experiment that we actually did for a customer, we had two different cell line clones, okay, and then we sequenced the alleles, and actually this cell line has 5 copies of the gene. But what we found was that each allele shown here, compared with the wild type, gave different kinds of deletions. Okay, so and all of these in each case gave frameshift mutations. But again, they're all different in length. Okay, and you can get in-frame deletions as well. We just didn't have any in this particular example. You can also knock a gene out using a donor plasmid. Remember, I had mentioned you can use donors for homologous recombination. And these are nice because you get the benefits of using both a selectable marker, if you're doing this in mammalian cells, as well as the fact that you can make a predictable mutation. Now you can say, I want to delete 7 base pairs in exon 1 in these exact 7 base pairs. You can encode that into your donor DNA This is a TALEN example, another technology, but you can do this with CRISPR as well, where you introduce both the CRISPR and the donor DNA, and then you select for the selectable marker. In this case, it's Puro, and then you get recombination between donor and chromosome, and you get a knocked out gene. Also mention how you can do mutagenesis or disease correction using homologous recombination. And in this example, which I mentioned in the table earlier, this is from a paper in Cell Stem Cell a few years ago, where this group used CRISPR and an oligonucleotide donor to correct a disease mutation that causes cataracts in mice. So the disease mutation is a single base pair deletion, okay? And so what they did was they took a single-strand oligonucleotide, which contains the wild-type sequence, injected this oligonucleotide with Cas9 mRNA and single-guide RNA into mouse embryos, and then they screened the mice, and what they found was, compared with the wild-type, they found that the mutation was corrected. And so they didn't correct individual mice of this mutation. They corrected the mouse line, but the mouse line was no longer passing down this cataract mutation to its progeny. So this was a very exciting finding. I also mentioned that one of the applications for genome editing, you can do in-frame fusion tagging. I did show a V5 example where a gene was tagged with V5. You can also tag genes with a fluorescent reporter, such as this application shown here. This is a project we did for a customer where we created a mouse line and we added a GFP tag. So the unmodified chromosome is shown here. We add CRISPR-Cas9 and then a donor DNA, which contains the GFP upstream of the natural stop codon. And then we see in the progeny, in the founders, the progeny, a GFP tagged gene. And finally, the last application I wanted to talk about for CRISPR, I didn't show this in the table. It doesn't get as much use or attention, but it is an interesting application, and that is targeted gene activation or repression. So again, most CRISPR applications that I mentioned use the CRISPR nuclease to cut the chromosome, and then that's the beginning event for things like non-homologous end joining or homologous recombination. However, there is another way of using CRISPR for targeted gene activation and repression, where you take a mutant of Cas9 that has no nuclease activity, and this can be fused to various transcriptional effectors, like the VP64 gene from herpes simplex virus, which is a transcriptional activator, or the Kreupel gene from Drosophila, which is a transcriptional repressor. So when these fusions are targeted to various genes, usually in gene promoter regions with a single guide RNA, you can turn a gene on or off without modifying the genetic code. So it would be an interesting way of, and a very useful way of modifying gene expression without modifying the coding region. All right, so now let's go into the section how we at Genecopoeia can help you with genome editing. So this is a basic overview of our genome editing products and services. We primarily provide plasmid clones that are designed for mammalian expression. And in this case, we will verify the sequence and deliver plasmid, usually in the form of DNA or bacteria. We also have a number of other products and services, such as functional validation. I'll go into that. And also the generation of transgenic mice, and I will go into that in a little while. Okay, so let's talk about CRISPR design and construction features. Again, this is in relation to delivering ready-to-use plasmid DNA. So we have various forms of the CRISPR-Cas9 nuclease. We have the wild-type Cas9 nuclease, and we also have various mutants like the nickase system. We have the point mutants that I mentioned before that reduce off-target activity, and we also have activation and repression systems. We provide expert target design for single-guide RNA targets. We do plasmid construction process of the highest quality. We provide quick turnaround time and advanced level technical support. This is an example of one of the CRISPR mammalian plasmids that we provide. This is known as an all-in-one plasmid. It contains the Cas9 nuclease or other versions of Cas9, as well as a single guide RNA. These are expressed under two different promoters. Cas9 is expressed under control, usually of the CMV promoter. We have other versions that are driven by EF1 alpha or CAG. And single guide RNA expression is driven by the U6 promoter. Okay. We also have what we term dual use or lentiviral clones. These are mainly used for cell lines that have fairly low efficiency of transfection. So instead, you might want to turn to lentivirus to generate stable clones, stable cell lines that express CRISPR-Cas9 and single guide RNA for doing your single knockouts. This is a two-component system, so we have we have different versions that express the Cas9 nuclease, and also a separate plasmid that expresses the single guide RNA. And again, this is a lentiviral vector. Now, these lentiviral vectors are also useful for single guide RNA libraries. There's a substantial amount of interest in using single guide RNA libraries. There are whole genome libraries out there from various academic groups like Feng Zhang's group or commercial entities. But we at Genecopoeia have developed something that we think is better, which is pathway and gene group specific libraries. We have 7 pre-designed single guide RNA libraries that target certain particular batteries of genes. Shown here is the number of genes that are targeted for each of these pathways and gene groups. But if you don't find something here that suits your needs, we can make to order a custom single guide RNA library. And the single guide RNA libraries come in a variety of formats. They're available either as bacterial stock, transfection ready DNA, or ready to use lentiviral particles. And these can be provided either pooled or as individual arrays. So you can buy these libraries from us with each gene separately rather than as in a pool. And so these are the advantages of our single guide RNA libraries. Our single guide RNA libraries are individually constructed and cultured in E. coli before pooling. That is, we have even for libraries where we have 1,000 single guide RNAs targeting different targets. These are constructed individually, and that is unlike most other companies that provide libraries where they're not even available as individual constructs. They're constructed as pools. And so this makes it possible to use these either as pools or as individual single guide RNAs. The pools are limited to 150 single guide RNAs per pool, which ensures excellent representation of each one. We verify the sequence of each single guide RNA, which provides high quality. And we have small library sizes, so this reduces the time and cost of screening and deconvoluting your results. So if you want to know more about the power of Genecopoeia's CRISPR single guide RNA libraries, you can read this technical note on the subject, and I will copy and paste that link into the chat box, and you can download that at your leisure as well. Okay, and I just sent that along to you. All right, so another area of CRISPR genome editing services that we have is for donor plasmid design services. So we will, not only do we provide single guide RNA targets along with Cas9, but we also will construct donors if you're doing applications that require a donor. And so donor plasmids are needed for certain applications like inserting a tag or doing a mutagenesis project. But also they're useful if you're doing even a knockout, because they give you the advantage in mammalian cells of having a selectable marker and a fluorescent reporter. So these can be identified easily, unlike with NHEJ mutations, which have no selection. So donors are great if you want to do a knockout in cells with low transfection efficiency. And also, homologous recombination mediated mutations are predictable, unlike NHEJ mediated mutations. So here are the features of our donor plasmid design and services. So we will design and synthesize the homologous arms that flank the given target site, and then we clone them upstream and downstream of a cassette that usually contains a selectable marker, drug selection marker, and a reporter gene. And these markers are flanked by LOXP sites for CRE-mediated removal if desired. So if you don't want us to design and construct a donor plasmid, you can do it yourself. We also provide our donor plasmids as cloning vectors with multiple cloning sites. These are some examples. Again, they come with antibiotic selection markers and usually come with or without a fluorescent reporter like GFP. So if you want to know more about Genecopoeia's donor plasmid design and construction services, you can read this technical note called Genome Editing, HDR Donors for Gene Knockout, Mutagenesis, Tagging, Safe Harbor, and more. And again, I will copy and paste the download link into the chat box for you. Okay, and I just sent that along. Okay, so the next product line of Genecopeias I want to talk about is known as Safe Harbor. So what is Safe Harbor? Safe Harbor refers to sites in mammalian genomes, and more specifically in humans and mice, that allow insertion of transgenes without causing harm to cells. So in humans, one of the most commonly used safe harbor sites, probably the most commonly used safe harbor site, is known as AAVS1, and in mouse this is known as ROSN26. And so the insertion of transgenes at this site presents no known adverse effects on cells. This is useful if you want to knock in for various applications that I'll go into. But also, this is an open chromatin structure, so it provides low copy number of transgenes and close to physiological levels of expression. So you get consistent and stable expression. So we have a number of kits that permit that allow you to readily insert transgenes at these safe harbor loci in humans and mice. The kits contain validated CRISPR reagents like single guide RNAs that are targeted to these sites, and you can pair these up with a number of different donor clones for knocking in various genes of interest. Now, one such donor clone product line we have is known as Safe Harbor Knock-in ORF donor clones. So as I mentioned earlier, very early in the talk, one of the powerful product lines of Genecopoeia is ORF clones. Okay, and so we carry genome-wide collections of ORF clones for humans and mouse. Well, the same is true for these Safe Harbor Knock-in ORF donor clones. We carry over 40,000 sequence-verified human and mouse ORFs that can be put into these Safe Harbor Knock-in ORF donor clones, and these ORFs are inserted between the AAVS1 or ROSA26 sites for ready Safe Harbor integration with our donor clone, with our kits. So what are some of the applications for Safe Harbor ORF knock-in? Well, there are several, such as you can use these to rescue a knockout or mutagenesis phenotype. So let's say you use CRISPR to knock a gene out. Well, you can put that gene back in very easily with using our Safe Harbor system by knocking in that given ORF to the Safe Harbor site. This is a classic way of doing genetics, molecular genetics, where you have a mutant and you want to rescue it with a wild type. You can also use it to, say, overexpress a fusion tag protein, or, and this is a very popular Safe Harbor application, you can use it to express a gene from a different species, such as you can express a human gene in a mouse line, for example. So if you want to know more about our Safe Harbor system, You can download this application note entitled Genecopoeia Genome Editing Tools for Safe Harbor Integration in Mice and Humans. And I will copy and paste the link into the chat box as well. Okay, and I just sent that link along to you. Okay, so the next topic I want to go over for our Genecopoeia gene editing services is shown here, and I put this title, "What do I do next?" And so basically, this is what you should think, what you need to do with the reagents once you receive them from Genecopoeia. We also refer to this as the downstream work following transfection. Now, for non-homologous and joining mediated applications, you might need to screen many colonies in order to detect modifications. For homologous recombination mediated applications, you might need to rule out random integration of the donor plasmid. But for all applications, you might need to mutagenize again or screen more clones for double allele or multi-allele modification. All right, so the first thing I'm going to talk about regarding this downstream work is functional validation. So here's a question for you. Why do functional validation? Well, for one thing, cell culture-based genome editing projects can take months, 3 months or more in order to complete. But not all CRISPR single guide RNAs are created equal, so we recommend testing the function of your single guide RNA first before undergoing long genome editing projects in order to avoid wasting time and expense. And that's because by default, the CRISPR guide RNA plasmids do not come functionally validated, but we do verify the sequence to make sure it is correct, but we don't by default validate them functionally. And so we recommend using a cell culture-based strategy known as the T7 endonuclease 1 assay. And the way this works, this T7 endonuclease 1 assay works, is that it's a PCR-based assay, and it takes advantage of the fact that, as I mentioned before, non-homologous end joining resulting from CRISPR creates mutations. And so when you do a PCR on a population of cells that has been treated with CRISPR, you get a mixed population if CRISPR was successful, where some of these products will have mutations and some will not, or some products will have a different mutation than a PCR product that has another mutation. And so the way this works is you denature these PCR products and then reanneal. And so you will then get another population of molecules, some of which will be homoduplex DNA, either mutant homoduplex DNA or wild-type homoduplex DNA, or you'll get heteroduplex DNA, which can be mutant over a wild-type or mutant A over mutant B. Okay, so when this assay is done, the way it works is you design primers using that anneal on opposite sides of the double-strand break, so upstream and downstream. And so you then denature this PCR product, as I mentioned on the previous slide, and then reanneal. And then the heteroduplex DNAs, if CRISPR was successful, are substrates for the T7 endonuclease 1 enzyme, which is a component of our indel check T7 validation and screening system. And so if CRISPR was successful, what you get is, as shown in this gel, you'll see 3 bands of predicted size. One is the uncut DNA, and this lane is the PCR product that has not been digested with T7 endonuclease 1. But you'll also get 2 other bands of predicted size shown here, marked by asterisks. And when you see this type of gel pattern, then you know that CRISPR is successful. And so we strongly recommend that people incorporate the indel check T7 validation system as part of their genome editing cell line workflow. Again, you do this at the start of your experiment in order to make sure that everything is working correctly. Now, if you don't want to do this assay yourself, we can do it for you. And so we have additional functional validation services. And this is an additional service that's available for both human and mouse single-guide RNAs. And what we do is we will transfect model cell lines for human, that's HEC293 or mouse Neuro2A, with your custom single-guide RNA plasmids. We will then do the same assay that I mentioned before, the T7 endonuclease 1 assay, and then we provide you with a validation report. So if you want to know more about the IndelCheck validation and screening system, you can download this technical note, and I will send that link to you in the chat box. Okay, and I just sent that along to you. All right, so the next topic I wanted to go over is copy number determination. So I have this question here: why do copy number determination? Well, and this is really only applies to cell line work, but for complete knockouts or mutagenesis, you might need to modify all alleles of your gene. Now, notice I said all alleles and not both alleles, and that's because not all cell lines, especially cancer cell lines, are diploid. Some cell lines, like HeLa, have 3, 4, or more copies of the genome. And so now you might expect multi-allele modification to be more difficult to achieve than single allele. However, that is not the case. At least one publication showed that multi-allele modification does occur more frequently than single allele. However, if you do have a cell line with more than 2 copies of the genome, you can get all possible combinations of alleles knocked out. You can have cell line clones with 1, 2, 3, or several copies and all possible combinations modified. So what I want to talk about here now is a project where copy number determination really does help you in your cell line work, CRISPR work. So this is a project we did for a customer, and I actually showed some of the data earlier. This project was to knock out the HDAC6 gene in the human MH7A cell line. HDAC6, histone deacetylase 6, is located on the X chromosome, and MH7A cells are human immortalized fibroblast cells. Now, when we took on this project, one of the things we asked the customer was, do you know how many copies of your gene are in this cell line? And because there was no information either on ATCC, where we got the cell line from, or in the literature, and the customer gave a fairly typical answer: they just didn't know. And so we then decided that to use fluorescence in situ hybridization, or FISH, to help us determine that copy number. And so we at Genecopoeia have a line of FISH probes known as vivid FISH that come in multiple fluorescent wavelengths to all 24 chromosomes. And when we did this experiment, we compared the fluorescence in situ hybridization patterns for the X chromosome between the MH7A cell line and a known human diploid cell line known as HT1080. So when we hybridized these cells with the vivid FISH X chromosome probe, we found that the MH7A cells contained, in most cases, 5 signals versus only 1 signal for HT1080. Now, there's only 1 signal in HT1080 rather than 2 because this cell line is male, as shown by hybridization with the Y chromosome probe, unlike the MH7A cells, which are likely female. They do not hybridize with the Y chromosome probe. And so this experiment, this result told us that this cell line has 5 copies of the chromosome that this HDAC6 gene resides on. And so this was extremely useful information for us to have, because it then helped us with the downstream screening efforts and sequencing efforts to identify clones that had all 5 alleles of the gene knocked out. And as you can show from this slide, there are indeed 5 different alleles: 1, 2, 3, 4, 5 in 2 different cell clones. Some of the alleles are actually represented multiple times because they actually represent multiple E. coli subclones. But both of these clones carry all 5 copies of the HDAC6 gene knocked out. And we also, in the process of screening, found other clones where there was 1 copy, 1 allele knocked out, 2, 3, and 4. So we, as I mentioned before, we did find cell line clones with all possible combinations of the gene knocked out. And so if we did not have the fish results that I showed on the previous slide, it would have been quite difficult for us to deconvolute these results. And so again, we strongly recommend using the Genecopoeia vivid fish probes as part of the genome editing workflow in human cell line, in mammalian cell line work. And so if you want to know more about using fish probes in a genome editing cell line workflow, you can download this application note, and I will copy and paste the link for that in the chat box as well. Okay, and I just sent the link to that application note to you. All right, and so that concludes the section on using on how to use CRISPR in mammalian cell line work. We also have a technical note known as genome editing in mammalian cells. What do I do next? And in addition, we have an entire webinar devoted to this topic. So I will send the link to this technical note to you in the chat box as well. All right, and I just sent that along to you. So feel free to download that at your leisure. So the next genome editing product I'd like to discuss is Cas9 expressing stable cell lines. So we have developed a large battery of cell lines that express Cas9 nuclease. In fact, we have the largest collection on the market. We have about 15 different cell lines that have Cas9 stably integrated into either the safe harbor locus that I mentioned before or randomly in the genome. And so these are stable cell lines that have constitutively or inducibly expressing Cas9. And we have, again, a large collection of pre-made lines, or you can have us integrate Cas9 into your stable cell line of choice. In addition, you can purchase a donor clone as part of our safe harbor system, carrying the Cas9 for do-it-yourself stable cell line creation. And in the case of the figure shown here, Cas9 is integrated at the safe harbor locus, which allows high expression of Cas9 and insertion without consequences. And this is ideal. These Cas9 stable cell lines are ideal for single guide RNA library screening or validation. Now, the next thing I want to talk about is CRISPR transgenic mouse services. I mentioned that we will create mouse lines that contain various CRISPR-mediated modifications. The features, we will basically do the entire project for you, the design, construction, and functional validation of CRISPR single guide RNAs. We also have design and construction of homologous donors if needed. We do injection of Cas9 single guide RNA, Cas9 mRNA, single guide RNA, and donor DNA into zygotes. We deliver at least 3 F1 heterozygotes. And again, this is a case study of such a mouse service that we carried out for a customer. I showed a variation of this figure before where we used CRISPR to tag a gene with GFP. So the customer had us tag the P2RX2 gene, which is a gene that's essential for proper hearing in mice. We used, again, the CRISPR system with a custom donor clone that we injected into mice carrying this GFP, and we delivered F1 mice carrying this GFP modification, and the customer was able to detect low-level expression, expected low-level expression of the GFP in the inner ear of mice, which is a very challenging thing to do. And so with that, I just want to summarize. I told you that CRISPR is a highly effective method for many applications from knockout, knock-in, activation, and more. I mentioned that the CRISPR-Cas9 is an RNA-guided endonuclease with some off-target activity, but it's very easy to design and use. And I also told you that Genecopoeia offers a comprehensive suite of CRISPR products and services ideally suited to meet your needs, such as clone design and construction, kits for functional validation and screening, Cas9 stable cell lines, and transgenic mouse services. So just before I take questions, I want to tell you about another upcoming webinar. We give webinars on genome editing often, usually at least monthly. And so the next one is entitled Genome Editing, How Do I Use CRISPR? This goes into the using CRISPR and downstream work applications that I mentioned before. This will take place on Wednesday, February 22nd at noon Eastern Time. This is the registration link, but again, I don't expect you to memorize or write down this link. Instead, I will copy and paste the link into the chat box, and then you can click on that link and register the same way you did for today's webinar, and I just sent that along to you. Okay, so now with that, I'm going to stop and take questions. Someone said thank you after I sent the first technical note, the answer, and so I would like to say you're welcome. Okay, so first question, do the Cas9 mutant work with all gRNA target sites, or do the target sites have to begin with a G? That's a very good question. There's some thinking that they have to start with G, and the reason is that in plasmids where you use the U6 or H1 promoter to express the single guide RNA, those single guide RNA sequences have to start with G in order to allow proper expression from the U6 promoter. And in one of the papers, they actually mutated a starting G to a non-G and saw a loss of activity. But so that would suggest that they do have to start with a G. And so what we don't know, though, is if addition of a G which is what we commonly use, causes a loss of activity. So for the time being, we recommend that people who are using the Cas9 point mutants use single guide RNAs that begin with a G nucleotide. Okay, so next question. Will you make the slides available after the seminar? Yes, we will post the slides on our website as long as a link to a video. Okay, next question. Would you recommend using an inducible Cas9 system? Well, that depends on what you're doing. It really is only necessary to use an inducible if you want to, say, maybe add a compound at a certain point and then knock the gene out or something like that. So it really depends upon your situation. Okay, next question. Which inducible system do you use for Cas9? Well, currently we use the TET system, so addition of doxycycline for turning it on and off. Okay, so does anybody else have any questions? Anybody? All right, well, if there are no more questions, I'd like to thank you very much for attending today's webinar. If you have any additional questions, feel free to call me at 866-360-9531, extension 227. Or you can email me at edavis@genecopoeia.com, and please visit us on our website, www.genecopoeia.com, and you can see all of the many different genome editing products and services that we have. So with that, I'm going to stop, and I'd like to say thank you and have a great day.